You're listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is May 11, 2012, and I'm your host, Dr. Jay Portnoy. Our topic today, application of genetic techniques. Our presenter is Dr. Marcia Chan. She's an investigator in immunology research at Children's Mercy Hospitals and Clinics.
Good morning, everyone. This is May the 11th. Is that correct? 2012. 2012. I'm glad I know that. We're here today for our COLA sessions. The first one today is on the application of genetic techniques, and we're pleased to have Dr. Marcia Chan here from our Pediatric Immunology Research Unit to give this talk. And thank you, Marcia, for agreeing to talk to us. And go ahead.
Thank you. Okay, so let's do our CME Credit first. Okay, so the first question is, restriction fragment length polymorphisms are variations in DNA sequences revealed by restriction endonucleases that cut double-stranded DNA into fragments of different lengths. True or false? Second question, microarray technology can identify potential targets as candidates for gene therapy. True or false? Third, diagnosis of CGD can be accomplished by A, DNA sequencing, B, RFLP analysis, C, ANV, or D, none of the above. Okay, so today we'll do an overview. We'll look at the methods to assess nucleic acids, such as the traditional methods of southern and northern blot. We'll discuss sequencing, PCR, and a microarray technology, and then we'll look at some of the specific applications of these methods to look at genotyping, maybe in newborn screening, and genome-wide association studies. So if we're talking about nucleic acids, when we're obviously dealing with DNA or an RNA, if we are looking at DNA, then we'll be looking at specific nucleotide sequences and looking at deletions or insertions or perhaps substitutions of bases. At RNA, we look at the level of expression, whether we're looking at upregulation or downregulation of genes. So for DNA, the common methods that we use are southern blot, restriction enzyme digest. We can sequence the DNA, or we can amplify it by polymerase chain reaction. For RNA, there's the northern blot, which is similar to the southern blot. There is reverse transcriptase, PCR, and then microarray analysis. So if we were to look at DNA, probably the most traditional method is looking at DNA by southern blot. And so once we extract DNA, say from a tissue sample or cells, then we want to expose it to electrophoresis, which will allow the different fragments of DNA to migrate according to size. And because DNA is negatively charged, it will migrate in this direction to the positive pole. And then we usually put some sort of a marker on it. We used to use P32 labeled markers that would allow us to determine the size of the DNA fragment that we were interested in. And then once we have run this gel, then we need to transfer the DNA onto some kind of solid surface. And this is what is really essentially the southern blot. And so we put the gel in a dish, but before that we have this salt solution. We have some kind of wicking system like a sponge. We put the gel on here, then the filter paper goes on top, and then we have more wicking material such as paper towels. And this usually, traditionally, it used to take overnight. And so that salt solution migrates up into the paper towel up here, and as it does so, so does the DNA. So that theoretically, all of the DNA gets removed from the gel and is now on this piece of filter paper. And so now we can actually examine the different DNA that we are interested in by hybridizing unique probes that used to be P32 labeled. Now we're using more fluorescent labeled things to the filter, which contains your DNA sample. And then we would allow this to incubate overnight and then wash it, and then we would expose our filter paper to X-ray, and then we'd expose the X-ray film. And so what we would have is our markers here, and then depending on the nucleic acid probe we use, we would see that light up in our DNA sample. So we could identify specific DNA sequences within our DNA samples based on the Southern blot method. Okay. Now we can also cut DNA using restriction endonucleases. These are made by bacteria. They recognize specific base sequences in double-stranded DNA, and so they cleave both strands of the DNA containing that recognized sequence. And the endonucleases will recognize either a series of 4 bases, 6 bases, or 8 bases. And here's an example of some of the commonly used endonucleases. Here's an example of 2 that are 4 cutters. And so here they recognize this 4 base sequence. For LU1 is AGCT and on both strands. And then here are 3 examples of 6 cutters, BAMH1, HIND3, and ECOR1. And they will cut at where the arrow is designated, but they recognize BAMH1 will recognize the sequence GG8TCC within the DNA. So these become useful tools to look for patterns of DNA sequences. So for instance, if we were going to set up a restriction enzyme digest, and we know that there is a BAMH1 site and ECOR1 site in this particular sequence of DNA we're looking at, we can then digest that DNA. Say initially, say with BAMH1, we would get 2 fragments from this 10 kb site. We get a 3 kb and a 7 kb. Now, if we were then to add ECOR1, then in addition to that 3 kb piece, that 7 kb piece would be cleaved into 2 pieces, 5 kb and 2 kb. And if you were to run this on a gel, then you can determine whether or not, say, your DNA sequence had an ECOR1 site by the resulting gel fragments you get after you digest them with the endonucleases. Okay. So using that technology, there is this, we can do what is called restriction fragment length polymorphism. And this will allow us to look for, say, deletions in a DNA. So for instance, here is a controller, say a normal person, here is a patient. In this particular segment of DNA, there are 3 MST1 restriction sites in the normal patient, or in the normal person. In the patient, there's been a mutation so that that 1 restriction site has been destroyed, and maybe the base has been replaced with another base, so that enzyme no longer has a site here. And so if we were to extract DNA from these 2 samples and then subject it to electrophoresis, do the Southern blot, and then probe with a region of DNA that corresponds to this particular gene, and then develop that blot on an autoradiogram, what you would see is that with that control sample, you'd have two bands, which would correspond to these 2 fragments. And with the patient, because it has lost that 1 site here, you would only see 1 single band. Okay, so that's what's called restriction fragment length polymorphism. Okay. So if you were in the lab doing this, what you would see, this is what your actual gel would look like. Here's your DNA ladder here, and there's our 9 patient samples here. And when you extract DNA and you just run it on a gel, because there are many different sizes of DNA, what your DNA looks like will be this big smear, so you can't really see anything. Once you transfer that to a filter into a blot, then you can probe that blot with that particular DNA species you're looking for. And now this is what you can see is the RFLP. Okay, and so in here you have a couple patient samples that don't have this particular gene, and then you have some that have that deletion. So here you have the two bands that you expect to see, say in this normal person, but these three patients have a deletion in that particular gene. Okay. So you can also use restriction fragment like polymorphism to genotype, and you can look for inheritance of RFLP markers. So say if your genotype looks like this, so your dominant pattern looks like these two bands, your recessive looks like this, and your heterozygous looks like this. You can take DNA from parents and siblings, and you can identify what genotype they have based on this RFLP. So you digest their DNA with a specific restriction endonucleases and take that probe that you're interested in, and then probe that southern blot, and then you could see that the parents are both heterozygous for this gene. You have one child that is homozygous recessive, you have one child that's homozygous dominant, and then you have another kid that looks like the parents that are heterozygous. Okay. So let's take a specific example such as CGD. And as you know, CGD arises from defects in any of 4 genes that encode this 5-protein complex, which is NADPH oxidase. 65% of CGD cases are caused by the mutation in the CYBB gene, and that encodes the GP91-FOX protein, and the remaining patients have mutations in genes for P22, P47, or P67. So if we look at the P67 FOX mutation, it is an autosomal recessive defect. It encodes the 67-KD phagocyte oxidase cytosolic factor, and that factor is required to form a functional oxidase. And so we can use RFLP as a marker to identify for, say, prenatal diagnosis of CGD. Okay? And so using that same technology that I just explained, here we have it with the actual family. And so what you see here is Dad. He is heterozygous. He has three patterns here. Mom is actually, she ends up being phenotypically normal, but she's homozygous recessive. Here is the fetus, who is heterozygous like Dad, and here's their particular patient, who is homozygous like Mom for recessive, but she actually is affected with CGD. And then she has 2 siblings, 2 brothers, who are heterozygous like Dad. And so in this particular screen, they were able to determine that the fetus actually had looked like Dad, and once that baby was born, he was normal, and his oxidase was functioning fine. Okay. So another way we can look at DNA is by sequencing it, and so we can actually look at the actual nucleotide patterns. And the most popular method is the dideoxy method or the Sanger method. And here we denature the DNA, double-stranded DNA, and then we use a specific primer for a specific sequence of DNA we were looking for, and we label that primer so that we'll be able to visualize it on a gel. And then the dideoxy method means that in addition to the polymerase and the nucleotides that you put in here, you put in what are called dideoxynucleotides. And at the 3' end of the chain is a hydrogen instead of an OH group, so that when polymerase picks up a dideoxynucleotide, then there's chain termination, so you can't get any more reading of that particular chain. And so when you set up this reaction, you set it up for each of your nucleotides, so you have a GATC tube. And so you have all the nucleotides in, say, the G tube plus the dideoxy G nucleotide. And then you run these reactions, and then you load them on a sequencing gel, which is a huge monster of a gel. And then you load the gel in a particular sequence. So in this particular example, it's GATC. So you would load it, you'd run it a little bit, and then you'd load more of that sample in that same order. And then when you develop the gel on a piece of X-ray film, you'll see these bands. And the way that you would read this sequence then is from top to bottom. So in this case, you have one that's A, C, G, T, C, C, G, T, A. And that would be your sequence of this particular DNA that you're looking at. So this is what it would look like on a film, and this is what you would have in this particular order is different. So you have A, T, G, C. And so this is an example here. If you started from here, you would have T, A, G, C would be the order of that nucleotide sequence. So that's the way we used to do it in the olden days. Now we have the automated sequencing system, and so instead of you actually doing this yourself, you can actually get it sent out, and it's very reasonably priced. So I don't think anybody actually does their own sequencing anymore. And so what the automated sequencing system is, instead of one, you have one reaction tube instead of four, and now all these deoxynucleotides are labeled with different dyes. And so you can read the sequence based on the colors. So if you were to submit your DNA sequence to a service that does this, then this is what the readout you would get back. You see all these peaks here, and the different colors correspond to the different nucleotides, and then they'd have the actual sequence up here that you could read. Okay, so again, taking the example of CGD, this time looking at P91 mutation, which is the majority of the cases where there is a defect in the sequence, and you get no oxidase. Less than 10% of the cases there are low levels of oxidase, and what they have been able to find out, there are 4 different mutations in the promoter of this gene that have been identified. 2 of them involve a change from cytidine to thymidine at position 52 and also at position 53 of the sequence. In this case, there's no oxidase activity in neutrophils, but there is oxidase activity in the eosinophils, which indicates you have some kind of cell-specific gene regulation of P91, and you get a mild course of the disease. If you have a mutation such that you have a change from a T to a C at position 55 or an A to a C at position 57, then you only have 5 to 10% of those neutrophils have near normal oxidase activity, and you have a severe clinical form of the disease. And so if you were to isolate DNA from the patient, select a primer that is close to where you're looking for the mutation, you can send these away for sequencing. And so here's the control sequence, and here's where the position 55 is, and where the normal person would have a T. These 3 patients have a C at position 55 instead of the T. And just so that you can get kind of an idea what that means. So here's position 55. So this is in the promoter region of the P91. So when we talk about a promoter, the position's always in the minus, because that's before the start site of transcription. So here's where that mutation is, and it turns out that's located in the consensus binding site for the S transcription factor. Okay, so the S transcription factor is critical for the transcription of the P91, GP91 gene. Okay. So in addition, we can also amplify the DNA by polymerase chain reaction. And when we do this, we don't have to use as much DNA. So we can use extremely small amounts of the sample. And basically, it's the same kind of theory. You'd have to denature the 2 strands of DNA, and then you add the specific primer that you're interested in. Then you get the elongation step, and you make a new strand of DNA. And then those double-strand strands of DNA are again separated, and then the primer comes back on. And so you can go for like 40 cycles and you'll get an amplification of your DNA sample. So here's an example of taking genomic DNA, and in this example they use 2 sets of primers, and this is an endpoint analysis, which means that, okay, so say after 40 cycles, this is the amount of DNA that's amplified. And so in tissue 1, you see that this particular tissue has none of that DNA that they're looking at. In tissue 2, with the 2 sets of primers, you see you have a strong signal here, but not as strong in the second set of primers. In tissue 3, we have an equal expression using both sets of primers. And with this type of assay, you always include a positive control, so you know exactly where your DNA would be migrating. And then we have a ladder here again, just to know what specific size you should have. And when you design the primers, you will know what the expected size is. And so that's what your ladder is good for. And then if you have a band that's way up here in one of your tissue samples, you know that there's something wrong with that assay, or there's something funky about your tissue sample, because you know what your expected size should be based on the primers that you select. Okay. So ways we can use PCR is in the detection of recombination excision circles. And so say if you were looking at development of T cells, you would look for these, what are referred to as TREC, or T cell receptor excision circles. And so in the thymus, as you recall, this is where T cell development begins. And so this is where the T cell receptor begins to rearrange its genes. And so you expect to get these excision circles. And so you can develop primers to detect these excision circles. And if you don't see them, then you know that there's something wrong with this particular patient. Okay, and that also is the case for B cell development. And these are called KRECs, for kappa deletion recombination excision circles. The same thing as the B cell receptor begins to rearrange the genes, you get these pieces of DNA that are excised. Okay. And so you can use this in the application for newborn screening for SCID and agamma-globulinemia. And this is just a chart showing, where you could use the TREC analysis and where you could use the KREC analysis for newborn screening. And this apparently is useful now, and you can actually extract the DNA from those dried blood spots that you get with the newborn, and you can pick up whether that kid's going to be SCID or has the George syndrome or agamma-globulinemia using this technique. Okay, so in addition to PCR, there's also what is referred to as quantitative PCR. And here the DNA is amplified using fluorescent dyes, and after each cycle, fluorescence is measured. And so here's what is called the threshold. And so a signal above this threshold is a positive signal. And you can get cycle thresholds here with quantitative PCR, and this is proportional to the amount of DNA is present. So whereas the other one is an endpoint analysis, so you're looking after 40 cycles, you can actually look at how much DNA you have that's amplified as the PCR is running. So say if you have a CT value of 18, that tells you that that particular gene is present more predominantly, say, than in a gene that if you had a CT value of, say, 25. Okay. So with quantitative PCR, we can look at single nucleotide polymorphism. And if you recall, SNPs are a substitution of one base for another at a single site, and it results from copying errors made while copying DNA sequence during cell division. And the SNPs can occur in a coding sequence, a non-coding region, or a promoter region of the gene. And so far, there's about over 10 million known SNPs in the human genome. Most have no significance, but others can alter regulation of the gene, such as gene splicing, maybe a transcription factor binding site, or they can result in altered gene product. And there are two types of SNPs. There's those that are called synonymous, which is a base change that does not change the amino acid, and usually these are the non-functional effects. And then there are those that are non-synonymous, where a base change results in a change in an amino acid or chain termination. And this is the non-synonymous ones are the ones that are likely to affect the protein product. And so here's an example of using quantitative PCR and doing SNP genotyping. And so here we have probes that have been tagged with two different dyes, so for each allele. So one allele is probed with a FAM dye, the other allele is probed with this TET dye, and then you run your DNA samples looking for the pattern. And so your homozygous wild types would cluster around here, your homozygous mutants would be here, and those that are heterozygous would be somewhere in the middle. So once you see a pattern like this, then you can identify which DNA samples are which. And then you need to confirm it by doing some kind of, probably by sequencing. And so here is the results of the homozygous wild type. You see here's the G, and you've got two Gs here, and you can see how high that peak is. Here's the homozygous mutant with two A's instead, and so here that's here. And here's the heterozygous. You see that the peak size is different, okay, so there's, which tells you there's only one G here, and then you can just see the A here as well. So this is your heterozygous population here, okay? So once you do the genotyping by RT-PCR, you always need to confirm it by some other method, and in this particular case, it's by sequencing. Okay. We can also, with DNA, we can do genome-wide association studies, or GWAS. And this is designed to identify variants in the human genome that's associated with a particular disease. And these variants can include deletions, insertions, and polymorphisms. They serve as disease markers, and they're associated with disease since the frequency of appearance is greater in the disease group. And so here's an example of a GWAS study. So you have your patient samples and you have your non-patient samples. You isolate DNA from your patients and your non-patients, and then you put them on where these SNP arrays. And so you have disease-specific SNPs and then the non-disease-specific SNPs. And so by the GWAS studies, you can associate single nucleotide polymorphisms with specific diseases. Okay. And then once you do that, then you need to, once you find an association, then you actually have to determine whether or not that SNP really is a functional SNP and really is associated with that disease specifically. Okay, so for DNA analysis and summary then, we can do a southern blot, we can do RFLPs, and we can genotype the DNA. An example is the CGD. We can sequence the DNA to look for specific changes in nucleotides, and I've shown you an example using the CGD example. And then by polymerase chain reaction, we can look at SNPs, or we can do newborn screening by looking at TREC and KREC. Okay, moving on to RNA then. RNA, we're looking at the level of expression which influences the function of genes. So we can look at either upregulation or downregulation of genes, and the techniques we would use would be northern blot or reverse transcriptase, RT-PCR, or microarray. And so the northern blot is basically the same as the southern blot, only you're using RNA. And I think the only reason they call it a northern blot is because the DNA was called southern blot for an investigator whose name was Southern. And so RNA is called our northern blot, protein's called a western blot. I don't know. And there's no eastern blot. So
it's not related to the direction
yeah.
that the molecules migrate.
Right.
Named after Ed Southern.
Yeah. So with the northern blot, we have our sample, but now we're extracting RNA instead of DNA. The RNA is separated by size, again by electrophoresis. We want to make a permanent picture of that RNA, so we have to transfer it to something that's stable, so it's fixed to a membrane. And then again, we want to, if we want to look for specific RNA species for a particular message, then we would use a labeled probe, and then again we would visualize this on X-ray film. Okay. So now with an RNA gel, unlike the DNA gel, what you would see are the two most prominent species of RNA in your sample, which is the ribosomal RNA. And so for humans, it's the 28S and the 18S. So these are the things that you would see on your gel. And again, you would have a molecular weight marker. So once that is blotted, then you want, if you're looking for a particular expression of gene, in this case we have a JERCAT and a smooth muscle cell RNA, we would probe it with a probe that would pick up this particular chemokine receptor, CCR8. And you can see that there's a lot more in the JERCAT than in the smooth muscle. In addition to that, you would want to probe this blot with what we call a housekeeping gene, which means that its expression is not changed. It doesn't get regulated. And so that would give you an idea whether or not you have actually put the same quantity of RNA in your sample. Say if you wanted to compare one sample from another, then you want to make sure you have the same amount of RNA present, and then this is your check to show that you've actually loaded the same amount of RNA. And in this example, you can see that the GAPDH is less in the smooth muscle cell, which tells you that you didn't load the same amount as in the JERCAT sample. Okay. We can also do reverse transcriptase PCR, and again, we can use much smaller amounts of RNA than we would use with a northern blot. And so in this case, with the RNA, we'd have to make cDNA first. So we make your first strand synthesis of the cDNA, and then you make a double-stranded cDNA, and then you can amplify it with the specific primers that you're interested in. Okay, and now this again is, so this is the endpoint RT-PCR. So this is after 40 cycles, and so if you were to run this on a gel, an agarose gel, your product, you might see something like this. So here is most gallbladder epithelial cells that have been simulated with two types of LPS, polysaccharide, one from E. coli, one from Pseudomonas, and they were looking at particular genes. So here they looked at a chemokine NIP2, and they've looked at TNF-alpha or IL-1 beta, and then again, of course, they have some housekeeping gene here, and they've used acidic ribosomal phosphoprotein. And so you see just by eye that these look quite comparable. There's no change in TNF-alpha, and we see there's a little bit of IL-1 beta expression. So if you wanted to quantify this now, what you would do is you would take a picture of your gel here, and now you would do densitometry, which is basically pixel counting. And so based on the pixel and the ratio of this to this, and say this to this, you can then graph it. And so you can see that your eyeball says these are the same. By densitometry, they are also the same, and same with TNF-alpha and IL-1 beta. And this may look this looks kind of like the same, but when you do the pixel counting, you see that with the LPS from Pseudomonas, it's a little greater in activity than with LPS from E. coli. So this is the type of way you can quantify this type of analysis. Okay. Again, you can also do quantitative RT-PCR and again look at CT values or the cycle threshold. And so this is just an example of, here's the GAPDH primers we use, and here's germline C epsilon. And you can see in some of these samples, for instance, in this sample, there's not as much germline because the threshold value is in, like, it looks like around 29, whereas in this sample, you know, the threshold value here is CT values maybe between 12 and 14. And then this here is just our control, which is a no template control. And now you can then graph this, and what you see is there's no germline epsilon in the non-stimulated samples, but with IL-4 and antibody to CD40 stimulation, we get a nice response to germline epsilon. But now if we add anti-IgE monoclonal antibody, we get a decrease in the expression of germline epsilon on the transcript. Okay. So we can also do microarray, and this is gene expression profiling, and the format is a chip, which is just basically a microscope slide, and it contains thousands of different oligonucleotides with unique gene sequences. And you can do cluster analysis, so you can group genes together based on the co-regulation and similar cell function, and you detect this with a laser. And so here's what a chip would look like here, and here's an Affymetrix chip. And so here in this one corner are a bunch of probes that correspond to genes that all have the same function or related function. And so you would put your RNA sample on here. Well, actually, it's a cDNA. So this is how you would set up your microarray. You'd have a sample A, say a non-stimulated, and sample B, which is stimulated. You extract RNA, you make cDNA because it's much more stable than RNA. And then you would label each of the cDNAs with a different fluorescent tag. And then you would add that to your array, and that would be hybridized to the array. And then you would, by a scanner, you would look at what you have. Okay, and this is the imaging, what you'd see. So because you use two different dyes, you can get some different colors. And so if in your sample A you have more gene expression in sample A than sample B, then whatever that dot corresponds to in terms of the genes, you have more expression in A than in B. If you have more B than A, then you're going to get a green dot, and if they're comparable, then you're going to get a yellow dot. And that way you can determine which genes are differentially expressed in your particular sample. And then once you have these results, then you need to confirm it by either northern blot or by QR-PCR. In this example, they've confirmed it by QR-PCR. So this is the microarray analysis. And here, these were mouse B cells that were stimulated with anti-IgM, and they also had some knockout mice. And so here's the microarray, and let's look at CD62 ligand. And so in the microarray, once those cells were stimulated, we see that in the wild type and in the BTK knockout, we had a downregulation of the CD62 ligand. And so we wanted to confirm that with QR-PCR, and we see a similar pattern of expression. Okay? So the microarray is great for screening, but you always have to confirm what you see with a microarray with another method. Okay. So the microarray technology in immunodeficiency disease, we can look at gene expression in patients with known defects, and it's an insight into the biology of the affected pathways. And it can also expand the understanding of the pathophysiology of the disease. And so such as the Bruton tyrosine kinase defect or the intravascular coagulation and fibrinolysis syndrome. And so if we look at the Bruton tyrosine kinase, we know it plays an important role in B cell development and signaling, and the mutations in the BTK can cause X-linked A-gamma globulinemia. And the microarray analysis revealed differentially expressed genes between the patients and controls, and this was published in 2002. And what they found in those genes were receptors, kinases, and transcription factors. And a couple of these kinases were FIN and HCK. And we can also use microarray as an exploratory tool to study differential gene expression in immunodeficiency diseases of unknown causes, such as CVID or selective IgA deficiency. And so both diseases are associated with impaired immunoglobulin production, and they both can lead to increased chance of developing autoimmune diseases and cancer. And so Chin in 2003 used the microarray analysis, and he was able to show that in CVID there were 179 genes that were downregulated and 46 that were upregulated compared with the normal control. And in SIGA deficiency, they were able to show that 68 genes were downregulated and 20 were upregulated, and these genes were similar to the CVID patients. And I won't tell you what all of those genes are, but the gene expression were skewed towards the Th1 pathway. Okay, So with RNA, we can look at differential gene expression. We can do that by northern blot, which is semi-quantitative. And then with northern blot, you obviously need more material than, say, with QRT-PCR. And you can, with QRT-PCR, it is quantitative in real time. And then with microarray, this is a large-scale analysis where you can see many different genes, whereas with northern blot or QRT-PCR, you really only are focusing on a couple of different genes that you're interested in. Okay, so let's return back to the credit. RFLP polymorphisms or variations in DNA sequences revealed by restriction endonucleases that cut double-stranded DNA into fragments of reference lines. True or false? Right. Microarray technology can identify potential targets as candidates for gene therapy. True or false?
True.
And diagnosis of CGD can be accomplished by DNA sequencing, RFP analysis, A and B, or none of the above.
A and B.
A and B, right.
Good question.
Sure.
In the clinic I do with Dr. Page, the eosinophilic esophagitis clinic, we are seeing increasing numbers of family histories of eosinophilic esophagitis. Nobody really knows what the genetic mechanism is, or if there is one. There's some reports of some association with certain genes and running in families and stuff. But when you were talking about like microarrays and you were doing the WAS or whatever you were talking about, the GWAS, excuse me. If you were trying to, if you had we have these patients and relatives, and if you were trying to look at that, how would you start that? I mean, would you, if you were able to get blood samples from these different family members or whatever,
right.
Is it to, get these blood samples from all these affected people and then try to get a bunch of normal blood from people that don't have these diseases and cross-match and sort of thing like those GWAS? Or
probably, because if you don't have an idea of the genes, the specific genes you want to look at, then you'd probably want to do a GWAS study.
So how difficult are those to do?
Actually, not at all. What you need is you get blood from the patient, and you'd isolate DNA, and then you would, actually Dr. Yi would help you with that because he has this whole lab that's devoted to do genome-wide association studies. So he has the machines that will do that for you. And my understanding is that all you have to do is provide him with the chip. And even if it's a clinical study, you may not even have to do that. I don't know. But for us, we just have to provide him with the materials, and then his technicians will do the rest.
So for the I guess the normal population, is that something that you possibly could get blood from the lab of serum that patients have that have blood work done?
Well, you'd have to have the cells, right? So, but for your normal controls, if you're not so much interested in age-matched controls at the beginning, and you just want to look at your patients plus just normal individuals, then you don't need.
You just have to give volunteers and normal individuals that are willing to give their blood.
Right, and you don't need a lot of blood to extract DNA. Maybe probably 5 mils of blood right in the cells. But yeah, if you wanted to do something like that, I mean, he has so much, all of the equipment in that lab.
I just think that because I went to the college meeting last fall, and they had a session on eosinophilic esophagitis. They had experts from around the country, and there were some people that were starting to do some gene analysis and thought they had some links or something, but everything is pretty nebulous. And but we've been asking about family history of any kind of GI disorder, and especially the last few months we've had. I think last month we had out of 5 patients we saw at one clinic, we had 3 that had more than one family member that supposedly was affected. So you start So, you know, if that's really the case, then you know, it'd be nice to try to you know, find out if there is some association.
Right.
You know,
do you know if anybody has looked at that in the literature?
I know that there's
Like, has done a GWAS?
I don't know if they've done a GWAS. I don't remember. I just remember that someone reported that they had done some research and they had some families that they'd looked at and they thought they had some. There was like one or two, in a sense, candidate genes that they were looking at or something, but it was just with a small group of people, sort of thing, that from their population.
Doesn't Hershey get DNA from everyone who goes to Cincinnati? Pardon? Like Hershey. Hershey in Cincinnati. I think she has this big DNA bank.
Yeah,
she? I don't
she
know
does.
if
I think she has a huge bank. But I mean, if you wanted to try to do a study like that, I would talk to her. Or I could talk to him on your behalf, whatever you like.
No, I should talk to Stephanie Page, who I do the clinic with, and
Right.
see what she says. But it just seems more recently we've just been finding more, you know, families that have the disease, and it'd be just nice to see if we could get any more data on these patients.
Right. Sure. Sure. But yeah, that would be. I think that if you're not sure the specific gene, then that is probably the way to go, is to do a GWAS. Because the microarray really is looking at gene expression, whereas the GWAS will look, you'll be looking at changes in the DNA. So whether there's an insertion or a deletion or substitution of a nucleotide. And then if you do that, then you could, if you're able to determine that, then you could do a microarray in terms of looking at gene expression. But I would do the GWAS first.
Okay.
You know.
Well, thank you.
You're welcome.
What is the cost difference between GWAS compared to if you're looking for specific SNPs?
Well, I guess it depends on what you want to do with that. If you want to look at a specific gene, then all you really need are the primers. And primers are not.
So would it be a lot different than if you're doing the whole?
Oh yeah, GWAS study is much more expensive than to do if you already know the gene. Yeah, sure.
Tara, did you have any questions? Good. Hello, Tara.
No questions.
Maybe she You haven't gotten to labor yet?
Not yet, no.
Okay.
Here. Same
We're expecting the baby this weekend.
It's worth knowing a little bit about how these techniques were used, even if they're not used now. Because number one, there'll be questions about them on different exams. Number two, you sort of get a better idea of how the biology of these things emerge. So the stuff about the CGD with the P91 promoter mutation altering the way ETS works. I mean, that just tells you that ETS is an important transcription factor in neutrophils, but not in eosinophils. So that sequence is either not important for eosinophil transcription of that protein, or there's other transcription factors unique to eosinophils that drive the production of that protein in the eosinophils. And it's possible maybe in those individuals their clinical syndromes helped by the eosinophils that make that oxidase. And that's why you can get different penetrations with the same kind of mutation. You can get altered biology that way. So some of these things are worth knowing about because it helps you understand how the disease, gets established and how it progresses. And plus, whenever the investigators figure something out, you're going to get questioned about it, whether it's in the ultimate and the critical important issue or not. If it's figured out, you'll be tested if it's published. So,
okay. Well, thank you, Marcia.
You're welcome.
We're going to take a 5-minute break, and then we have a guest speaker, Dr. Jacobs, who will be talking to us. We're going to do an immunology case conference. Do you want to report it?
This has been an ACAAI production. To learn more about Conferences on Lyme Allergy or the American College of Allergy, Asthma, and Immunology, go to www.acai.org. See you next time.